same time throughout the week. And today, because it's our first day, we have not only the speaker, but let's start with welcoming the assistant director of this program, Drew, who is going to give a brief overview about the program and talk about TAMIT's, this ambassador program, and the water summer school before we jump into the next. So, Drew.
Hi, everyone. Thank you, Devashish, for putting this on. I am Drew Casey. I am with the Institute of Data Science at Texas A&M University. We are a university-wide institute that was created between the university and then two state agencies, the A&M Engineering Experiment Station and A&M AgriLife. So our mission basically is to support and grow data science, AI, and machine learning across the university. We accomplish this through training and resource generation, research support, faculty support, all different types of elements. And we work both internally and externally. For students, we have a lot of engagements, including our ambassador program, which Devashish is one of our ambassadors who helped organize this event. We host almost weekly seminars or tutorials, the data competition every year. We even support other data competitions, including TAMU Datathon, which is, I believe, open to everybody, not just A&M students. And we have a lot of academic programs aimed at both students and faculty. We have course development grants, research development grants, and then for students, travel grants as well. And if you're an undergraduate student and you're thinking about getting a master's, why not a master's in data science? So we have our data science program. It's a 30 credit hour. You complete it in 3 semesters, so 1.5 years. And you can customize it based off of your interests. There's 4 core classes, and then you take 3 elective classes based on your track, and the 3 elective classes, whatever you want. And applications are accepted each fall for the following fall, so about a year in advance. And one of the best parts is that we have a competitive scholarship program where we offer $1,000 to first-year students, and this allows them to get in-state tuition. So if you're out of state for whatever reason, it saves you more money in the long run. So if you're interested, you can go to text.ag/MSDS, or you can scan the QR code on the website or on the screen. And like I said, we have our ambassador program. They put on great activities like this hydrological summer camp. We support data science competitions. And so there's a lot of engagements for students. And then if you're a researcher out there or you're wanting to do research, we also have our 8 thematic labs. They do all sorts of visualization, that database, AI, large language models, whatever you can think of. Each of the labs has a theme, a general theme that lab focuses on, but their research crosses across disciplines. And then we also have our affiliate program, our industry affiliate program, that they work with us to develop resources and tools, and they provide a lot of funding for activities like this. Our industry affiliates help sponsor the ambassador programs and help support that. So I'd like to thank them whenever I can. And generally that's it. But if you have any questions, you can go to the website. It's tamids.tamu.edu. It's there at the bottom, and you can find our contact information. You can reach out to any one of us. So you can reach out to me, Drew Casey, and look forward to the rest of this week. And again, thank you, Devashish, for putting this on. Uh, and if there is nothing else, I guess I will hand it back over. We can get on to the main speaker for today.
So again, before getting started, I'll make sure that hit the record option. Um, with that, let me give a brief introduction of today's speaker. I'm so happy that Andrew is here. I have known Andrew for a couple of years now and have seen his research work and read his research papers. So Andrew basically finished his PhD at MIT. He was working with Professor Dara and Takabi. And then he moved on to work as a NASA postdoctoral program fellow at Goddard Space Flight Center. Right now, he's the assistant research scientist. His research interest is on how climate variability affects water and carbon cycle responses. And without further ado, I'll request Andrew to begin his presentation.
Yeah, thank you, Devasish. Appreciate it. Thanks for the invite. Give me one moment to find it. Yeah, so hi everyone. I appreciate the good introduction from Devasish. Pretty thorough, and yeah, I'm going to talk today about, I think, a little bit more of an applications. I think the next couple speakers are going to talk a little more generally about some methods and kind of how to think about different parts of remote sensing and AI and machine learning. I think that's really interesting. This is going to be a kind of a satellite assessment with kind of a neat data set. I think it's going to take me a few slides to set up the significance of it, because this title is so trivial sounding, and I admit that it may not sound interesting at first, and I don't think it initially did to me when I started working on this project with my PhD advisor a couple of years ago. But I'll just say, one thing I'll say is, as I've learned over the years, this global vegetation response to rainfall events, I think we all think it's kind of a simple thing. Okay, rain falls, and what does a plant do? I think once we actually start to think about what a plant does after a rainfall event, it actually isn't so trivial, and we actually don't know a lot about that. Even for if you water your house plant, what happens? And so such a simple sounding title actually is quite complicated, and it actually it might not sound that important, but it actually scales up to be quite an important thing. And so that's something I've been studying for the last few years, since about 2017 or so. And it's kind of just interested me more and more each time. So you might look at this title and not think it's that interesting. And I hope by the next couple slides and maybe by the end, I've convinced you there's something interesting here. So yes, as Devasish said, I'm a research scientist. I'm at NASA Goddard Space Flight Center. I'm sitting in Maryland right now. I'll go a little bit through my background. I grew up in Allentown, Pennsylvania, kind of in the middle of nowhere, and I ended up moving into Philadelphia to Drexel University, into the city, which was a big culture shock for me to go from really the middle of nowhere into the city. But now I've kind of lived in cities and in really populated areas since, and it's kind of shaped how I've, the two different things of middle of nowhere and the city has kind of shaped how I've thought about the world a bit, and it's been very interesting. But I started in civil engineering, and then I moved into hydrology, and when I went to MIT then in grad school, it became a science mostly. Even though my degree is civil engineering and my PhD, it's really science, and it's a really different way of thinking, and it's not fully applications based, but really very upstream. How why does the world work the way it does, rather than let's find a solution to a problem. And then from there, I really learned hydrology with Dara. Dara was an amazing advisor. It was quite a difficult program, but also extremely fun. And I got to learn about SMAP, and I'll talk about that in a second. But then I moved from hydrology, then when I moved to NASA Goddard, into the carbon cycle, and working with Ben Poulter. Ben Poulter's actually at the White House right now, of all places, working on a greenhouse gas committee under the Biden administration. And it's been really awesome working with Ben, because he's a carbon cycle expert, and I've really been able to tie the water and carbon cycles together and how they're linked, and really think about that. And Ben doesn't even know a lot about hydrology, so it was kind of neat to bring a different expertise to the lab. And now I'm kind of on my own as a research scientist, which has been quite fun as well. And actually, I'll step back. What's really funny, that I've I've been in the Northeast, but this line I just noticed is I-95, Interstate 95. I've lived like really close to I-95 exits. I'm just realizing that now. It's kind of funny that you move quite a big distance, but I'm still on the same highway. Um, um, yeah. So what are what are the things that I think about now as a scientist? I'd split them into three different things. One of them is really I think in a plant physiology framework. How do plants work? I'm not a true plant physiologist because I don't really go into the field and study these things. I study these things from space, and it's really hard to tease out causality of what's driving plant function from space. But there are a few things we can do with satellite instruments at this in really large scales. And so I'll talk a little bit about how climate drives plant interactions. But a lot of it's land surface hydrology still. A lot of this at the end of the day is a land surface hydrology background. And I'm plotting this evaporation versus soil moisture curve here. It's so funny because all of the problems that I work on seemingly come back to this curve of this black line of evaporation versus soil moisture. And it just every problem that I think is interesting and new, it always comes back to this darn curve, this black line here. And this problem I'll talk about today partly comes back to that as well. But yeah, I kind of have a hydrology and ecology background, and mixed in with understanding some of the remote sensing physics and why and how to retrieve certain properties from satellite measurements that give you some type of emission or some type of scattering parameter, and you can turn that into something interesting about the surface, such as vegetation water content. So I've studied a little bit of the science side and even on the algorithmic perspective in remote sensing. And as Devastation said, I think a lot about variability rather than the mean change. I'm trying to understand how does the land surface respond to variability mostly. And I'm sitting here talking to you from a heat wave in Maryland right now. We've had a heat wave the last week or so, and it's kind of continuing into this week with pretty high temperatures for us. And a lot of this is that we're seeing climate change signals now, and these are very big signals. It's not going to be we're not going to see things in the year 2100 necessarily, that we don't have to wait till year 2100. We can see a lot of these signals now. And I'm trying to understand what physical mechanisms are driving these surface responses. How does heat wave shape plant responses? How does drought shape plant responses across the globe? And one of these in particular, the variability side of things, and I'll talk about this a lot today, is how more intense, less frequent rainfall is impacting photosynthesis and plants. The hydrologic cycle is intensifying. You might have heard about this. The specific feature I'll talk about here is the daily variability of rainfall and how when it rains now, it's becoming a lot stronger, and how that's shifting plants. And with that question, the variability of and its impact on plants, interstorms become a big factor. And it kind of comes back to this idea of just individual rainfall events. Understanding plant responses to individual rainfall events becomes it also a lot of the times becomes the answer to the problem. If you can understand that fundamental timescale, you can understand a lot about the variability of plants and how the carbon cycle and how crop yields are responding to rainfall. And drylands inherently emerge as an interesting location for variability. So this is a lot about me. I've spent too much time there. What I'm funded on right now is not stuff I will talk about tonight, but I'll mention briefly that I'm a member of the EcoStress Science Team, which is a land surface temperature instrument on the International Space Station. It's a 70-meter land surface temperature product that it generates. And then I'm also co-leading a dryland field campaign scoping study. I'm not going to talk too much about that tonight, but you can email me at andrew.feldman@nasa.gov if you have any questions. I'll do my very best to get back to you. I'm quite swamped with that second project right there, this arid project. But I'll do my very best to get back to you, and you can find me on Twitter at _Feldman24. There's a long story behind the 24 that I won't get into, but it makes sense. So what I will talk about today is this article that I wrote in 2018 with several awesome people, one of which was my PhD advisor, who actually gave me the idea when he had a fever one night it was a winter February, kind of was, I think, sitting up in bed for a couple of days and actually dreamed up this idea that I thought was actually, I told him I thought it was a bad idea at first. And here I am 7 years later studying the same thing. And I will say that he was right. And I will not be afraid to admit that. And the cool feature of this is it sounds, again, very trivial. And the title here is, you might not make sense of it, but I'll explain it a bit. It's just really how plant water content is responding to individual rain events and using microwave remote sensing to get at this. And microwave remote sensing is so interesting because it gives you water. It gives you information about water. So if we all were able to see in the microwave spectrum, we would see, how wet things are. So humans would be pretty wet. We're made up a lot of water. We have a high water content. So that would be a cooler signal. But if we looked at a very dry wall in the room that you're in, it would be a hotter signal. It would be drier. So that's kind of the idea behind microwave remote sensing, is we're able to see water if we tune to that longer frequency. So what is this context of this problem I'm thinking about? I mentioned this before, that there are becoming to be fewer larger rainfall events. And again, as I said, we for the hydrologic cycle, a lot of the climate change discussions we're hearing is, how wet will it be in year 2100? Will it be too dry? Will the Colorado River, completely be depleted by 2100? A lot of what I'm talking about here is we're seeing these signals right now about daily rainfall variability. This is a signal that's pretty strong, and it's now even acknowledged in the IPCC, the climate reports, that we're seeing fewer but larger rainfall events. And everywhere where it's yellow here, this is showing model projections actually into 2100. The model 28 climate models, CMIP6 models, agree that there's fewer larger rainfall events. And there are going to be fewer larger rainfall events in the future. I can show you this exact same plot, a very similar plot with observations between 1980 and 2020. So observations are picking up on the signal, and a lot of the surface here is yellow. So what it means is that it's not raining as much. It's not raining as frequently. So the wet day frequency is going down. The rainy days throughout the year is becoming less and less. But when it does rain, we're seeing a higher intensity of rainfall events. And this is a pretty strong signal, and there's actually well-explained reasons behind it that are actually attributable. And if a lot of you are in Texas right now, I'm sure that even in the thunderstorms you're seeing today in 2024 are actually stronger on average than what you saw in 1980, because the signal is pretty strong in Texas as well. And there's actually less consistent trends in the total annual rainfall amount. That's not as true for the Southwest U.S. The Southwest U.S. is drying out, and some of the Great Plains are drying out. But across the globe, it's actually pretty mixed. We're seeing a little bit of a wetter signal on average, but it's mixed, whereas this signal here, more intense, less frequent rain, is pretty consistent. So what does that mean for plants? How do plants respond to individual rainfall event changes of larger rainfall events and longer dry spells? It turns out they're competing effects. And by the way, I'll stop a second here. Debasish, if I'm missing anything, or if something wasn't clear, or if I should elaborate something more, I'm trying to follow the list of topics to cover. But if I miss something, please just interrupt me. It's not a big deal. If I'm talking too long about something, please go ahead and interrupt me. And anyone is actually, please just ask questions anytime. Yeah, and I'll get to Python code soon. I'll get to maybe some fun stuff there. So I'll make sure I don't ramble for too long. So
Andrew, just if I can, I think it's worthwhile maybe explaining even the simple definitions of winter storm, because there could be people other than hydrology. So, I was just
no problem.
thinking.
Yeah, thank you. And actually, this is the perfect picture to do this. So the dashed line there in the background, so the x-axis here is time in days. Each little tick is a different day. The dashed line there is supposed to show kind of current climate conditions of, it rains, soil moisture goes up, and then after a few days, it's drying, and then it rains again, it goes up really fast, and it dries. Interstorm is the time between two storm events. It's the time that there's a drying of soil moisture. So if it hasn't rained in a few days, you're in a dry down right now, you're in an interstorm, you're waiting for the next rainfall event. And then when that rainfall event comes, the soil moisture pulses up to some higher value, and then it slowly declines over a certain number of days. So does that make sense?
Yes.
And the idea here is about these climate change signals I'm talking about that we're seeing in observations, and models are projecting them pretty consistently, is that when it rains, we're going to see a bigger jump, a higher soil moisture after that rainfall event. But we're having longer dry spells then. If we're having less frequent rainfall events, that dry spell is lengthening out. We have more time for soil moisture to go to really low values. So you have these competing effects. You have these big rainfall events, you dump a ton of water on the soil, but then you have a long time until the next rain event on average. And so soil moisture might be drying to lower values than what it normally did. So we're comparing these dashed lines to that black line there. And this is the problem I'm talking about. And so I'm arguing here, to understand this problem, we need to understand plant responses at interstorm timescales. That's a pretty critical timescale, and it makes sense. I guess it's almost intuitive, but we haven't thought about this much at all, really, as a community. As even in plant physiology, people have not really thought about what happens when it rains. Maybe to a satisfying degree. A lot of the focus is on seasonal timescales. When do leaves come out on trees? When do the leaves start to die and fall off of the trees? It's a lot of seasonal timescale thinking. This short timescale hasn't really been thought about. So I'll say my first question here for the group. Maybe just to think about this a bit, how would larger storms but longer dry spells influence plant function? Would we see no change because the longer dry spell balances out how much a larger storm, how much a larger storm affects plants? Or does the larger storm effect of more water, do plants just love that? And so we see an increase in plant function throughout a year. So this is maybe the question can be across the year, annual plant, the annual mean plant function. Do plants like larger storms but longer dry spells, or do they not like that, or is there a mixture? So please put your comments in the chat. If someone, use A, B, C, D, and if someone puts an A or a B or something, maybe upvote it so it's easier to count. Okay, so a lot of C's, people think that there's going to be a decrease because the longer dry spell outweighs, or a mixture. Some plants will like it, and some plants not like it. So what's the answer? Okay, well, it turns out that there's widely varying responses. It's actually not very clear. So the right answer here is D. But if you answered A, B, and C, that's true in some places. So I guess the most correct answer is D, but if you answer something else, it's true somewhere. So what this was actually a review I conducted with several others, and just published earlier this year. And we went through and grabbed all the different studies that looked at how plants respond to fewer larger rainfall events. This was a meta-analysis, went through back to the literature. And what we found was that dry places, so places that receive less than 500 millimeters of rainfall each year, pretty much west of Texas is like that. You get to central part of Texas and you go west all the way to California. That's less than 500 millimeters of rainfall a year. Those plants tend to like fewer larger rainfall events. They tend to like that larger rainfall event. But some places don't respond. Some of those plants don't respond at all. Whereas wetter locations tend to respond negatively. So, some of this is thinking about why do they not like the large rainfall event? They tend to really not like that longer dry spell. The reality is, it's a very uncertain thing. And I want to capture the uncertainty of this. This has been studied in a few little locations at these different points. These are different places that people did field manipulation experiments. They actually put a plot out and left one plot alone, and the other plot, they changed how frequently it rained and how large the rainfall events were. So you get a very limited perspective of the globe. So, this panel on the top left here that I'm presenting, this is the average over a couple sites and maybe a couple regional analysis, but we don't really have a good picture across the globe. And so, this answer is uncertain, and we're still very much studying this, and it's something important to study. We have from these studies, they've mentioned that the annual rainfall changes can be between negative 30% to positive 30%. So pretty large plant function changes. Crop yields can change from this quite a bit. You can have up to 30% change in function throughout the year for crop yields and natural vegetation's amount of photosynthesis and carbon uptake. So this is not a trivial thing. And what I'm arguing here in some of my research, and toot my own horn, that interstorms are the answer. Thinking about individual rainfall events are the answer. As simple as going to that house plant of yours and pouring water in it. What happens? So I'll step back. This is my academic grandfather, Peter Eagleson. He actually died, just passed away right before I presented my thesis defense. And Peter Eagleson studied interstorms actually for years. And his argument, so I'm not completely doing this out of the blue, his argument was that to understand evaporation throughout the year, to understand soil evaporation, he theoretically modeled that if I understand the physics of an individual rainfall event, so top of this here is just a model of arrivals of different rainfall events. You have a big rainfall event in a short period of time, and a long rainfall event, but not not very intense. He said if you can model these interstorms, if you can really understand what the evaporation physics are happening, you can understand evaporation over the full year. This is the building block you need to understand evaporation over an entire year. And he showed this with theoretical equations. They didn't have a lot of data at the time, I think, when this was written in 1978, to show this more widely. So proving this with a theoretical model, he was able to show that the interstorm is actually a very important unit of time, and you can really scale things from that individual rainfall event. So this is my advisor's advisor for PhD, and this is something I've kind of carried on, I think, from his legacy. So part of this is to say the things that we do as scientists, they kind of might seem to go into an archive and be pushed away, but there's people that pick up what you do and they carry it on. And so this is the idea behind science, is to kind of learn new things, and then people that come after you pick it up and carry it on from there. And so I kind of encourage that this happened in this case, and this might happen to you as well for whatever you do. So let's think a little bit about this problem again here about individual rainfall events and your house plant. If you were to go and dump water on your house plant right now, how rapidly does that water go into the plant? So let's think about this a little bit. I bet you haven't thought about this much, and it might have been a simple question before, but it actually isn't that easy of an answer. So how fast is it? How fast does water go into a plant? A, B, C, D. Again, depends on how stressed the plant is. I love that answer. Okay, so yeah, the answer is it depends. I'm actually going to jump to this slide first. There's something called the RC timescale. If you go into the plant hydraulics literature and you make a plant hydraulic model like this on the right, I won't have to go into the terminology here, but it's basically modeling how water moves from the soil into the xylem, which is the conduit within plants, up into the leaf and then out because of transpiration. If you do this, if you model all these components and you put in reasonable values for how much resistance there is along the pipeline, it turns out that it takes a short amount of time. The answer is it's short. If there's well-watered conditions, and this y-axis here, so I'm going to show this is what the model produces if it's well-watered conditions. And actually, the 0 days there this means it's kind of hours, but the y-axis here is how much water is stored in the plant. So lower values, more negative values, means there's less water in the plant. And at time 0 here, it's pulsed up to the soil is pulsed up to basically really wet. It's a lot of rain on the soil, and you can see pretty quickly the plant picks up the water. So this is minutes to hours. Usually you're talking about an hour, 3 hours or so is the answer in well-watered conditions for most plants. That's most plants to, a cacti, big storage plants like cacti have a lot of storage capacity. Sequoias in California, a ton of storage capacity. It might take longer to fill that up. That might be a couple days. And yeah, here's a couple examples of this here from field experiments to show. And this is one here on so June. I think this is Arizona, Danielle Mace. This is in Arizona. This is in Tucson. So this is I think this is a grassland site, and they measured the water potential, the hydration status. More negative means less water. So it was, time before the storm there. It's pretty dry. It rains, and then it goes into the plant pretty quickly. Within that day, overnight, it went into the plant. So pretty rapid response, sometimes no response if it's well watered. I'm actually going to jump here. Actually, I put that the next question too early. Sorry about that. Jumping to another slide. This plot here shows a plant that took a while. Oh, never mind. That's not right. I don't know where I put this. There it is. Why did I'm very sorry, folks. I moved this slide way too far. I knew it was here. This is an orange tree. It was in very dry conditions. I don't remember where this orange tree is. I think it might be in the Mediterranean area. I think this might be in Spain. This tree took a couple days up to a week to rehydrate because it was very dry. This is very dry conditions in some of these, and it took a couple days. It just rained one day at day 0 there. Took a couple days. So it depends. The answer is it depends, but it's typically like an hour, couple minutes to an hour for typical plants in your house. That's the answer. So I'll step back here. Now, my next question is, can a single rain event cause plants to grow? And when I say cause plants to grow, I mean in a statistically significant sense that we can tell that there was growth to some degree, greater than 5% growth, or sorry, greater than like 1% growth above the noise. Yes or no?
You can go ahead.
Okay. Yeah, I'm going to do my very best to walk through this Python code. If you were wondering why I chose to do some very silly thing with a function, it's probably because I'm not a very good coder. I actually learned everything in MATLAB, and then I switched to Goddard Space Flight Center. And the U.S. government, at least the agencies that they don't want to fund, are quite poor. So NASA doesn't get a lot of money, and we don't get MATLAB licenses, which is pretty consistent. So I'm using a free software, which is Python. So I had to learn Python very quickly within my first start date of my postdoc. And so if I have bad coding, I'm sorry, but you'll be able to use the code to you'll be able to use the code and play around with it. So something that's key about Python, if you use MATLAB, that you don't have to do in MATLAB, is Python. You have to load all of your libraries. So up here, I'm just loading in all of the different functions that I might use. I think probably the most important one for working with data sets is NumPy, which is a kind of matrix library to do functions with matrices. So all the data sets that I really play around with are these 3-dimensional XYZ, latitude, longitude, and then time variables. So what I've included and what Debasish will send out to everybody is I included a small folder that is this script here. And the only thing you have to really change is this the directory that you saved the data in, because then I gave you also a sample data set. If I try to give you that whole data set that I showed you in that Zenodo file there's, I think, 30 gigabytes of data. If you try to load all of that at once, it will certainly crash your computer. So don't do that. I'm just using a snippet of data, and it's just in Africa. It's just a piece of Africa. I think this is in Sudan, a grassland in Sudan. So dry grasslands. We'll really see the pulse reserve behavior here. So I'm loading the data. I'm just going to go through each step sequentially. I'm really sorry if you're not able to see my screen that well. Is there a way to zoom in? Do this. Nope. Okay. I'm not going to play around with that. Load the data. I'm going to jump through. So the function that this script will perform is it's going to look at individual pixels that I've given you. There's a soil moisture data set here. So this is soil moisture. And then this is that vegetation water content data set. I have the latitude and longitude references. I have date. So we won't use all of this, but I'm going to first jump to 1 pixel in Sudan. So I'm going to select in this sample script here. So I'm going to actually show you this data set here, this VOD, or vegetation optical depth. It's the water content data set. It is a 2000. This is because it's 2000 days long. So this is 7 years of data over a 10 by 10 pixel area in Sudan. So there's 7 years of data in this. So I'm loading just one of those 10 by 10 pixels here. I'm loading the soil moisture time series, and I'm loading the vegetation optical depth time series. So if I plot the vegetation optical depth time series for you real quick, then I'll look, I might not do it. Oops. It's going to look like this. Can you see that? Did that work, Dennis? Can you see my a plot there?
Yes.
So yeah, this is vegetation depth over 7 years. You see, well, it's actually 6 different seasons. You're seeing 6 rainy seasons here between 2015 and 2021. So you can see the peak up here when it's the middle of the wet season, and the low down here when it's in the dry season in Sudan, in Africa. Okay, so now what I'm going to do is I'm going to plot soil moisture and vegetation optical depth together. What do they look like if I plot them together? So this shows that very common curve I showed you before about evaporation and soil moisture. It kind of shows a little bit of a kind of a broken stick model there. That when it's really dry, vegetation optical depth, or the water content in the plant, is very low. But as it gets wetter, it goes up and up and up, and then it levels off at something, or it becomes noisy. There's other factors that control the amount of water in the plant above a certain limit there. But you can kind of see a little bit of a 2 regime behavior there. Low soil moisture, it's very linearly related with water content, and above that, it levels off. So let's jump back. So now we're going to analyze the data. Now we're going to go through and pick out when it rains. So we're going to look for dry downs. So what this script does from here is it goes through that data set, and it actually looks through the soil moisture data set. So I'm going to plot the soil moisture data set here. I'm just going to show you the example time series. This is the soil moisture data set, so you can see the 6 wet seasons. It's going to go through this and find all the storm events in here. So I'm going to zoom in a little bit. I know I'm doing this pretty rudimentarily, but this is how I play around with Python. And it's going to go through and find when soil moisture goes up, and then when it's drying for a couple of days. So it's going to go through and find all these events of soil moisture going up and then down from a rain event. So it's going to find all of the peaks and all of the valleys in soil moisture. So I'm going to initialize. These are a bunch of different variables, a bunch of different empty variables there. I'm going to convert the water content and soil moisture into a different name, so I save it as something else. And then one of the things I'm doing right here, I didn't comment this, I'm sorry I didn't add a comment, but this right here is going to take out only the, it's going to take out all of the NAN data. So there's some days when there's no soil moisture value because SMAP did not pass over that Sudan on that day. And it's going to get rid of those NANs, not a number. And it makes our analysis and performing functions in Python a little bit easier that NANs are out of there. And so now what this does from here is it takes a little while to explain some of these, so I'm going to kind of give this to you in simple terms. But what this does is it's going to find this NumPy where function. It's going to find all of the cases when soil moisture went up to a peak. That's what this is, IPB. This is the peaks. And then the troughs are the low points here. These are when soil moisture comes down to its valley. So it's going to find all of those peaks and valleys there. I'm going to show you. Let me see if this, actually, I'm not sure if this works. It does. Okay. So here's one example of a peak and a valley. that it found. The time between a peak and a valley. So it found one peak and a valley. This is the 17th peak and valley that it found. And so this is one case of soil moisture going up to some peak after a rain event, and then it dried down over probably 10-day period there. And then this down here is at that same time, the vegetation water content data set, what that's doing during that dry down. And you can see there that it is going up during that, after that rain event. So there's an example there of it rained. So that's just one of the peak and valley sequences that it found. So I'm going to loop through. So what this does here, and I'm sure you can find a way to reduce this down to a couple lines less. But what this loop does, it's going to loop through and find each dry down event. It's going to find each of those peaks and valleys. It's going to put them together and say, how often was there a peak and valley that lasted a couple days, basically? So it's going to find all those soil moisture dry down events between rainfall events when their soil moisture is consecutively decreasing, and it's going to log them into different variables. This the soil moisture dry vec is the soil moisture dry downs, and the vegetation water content during the dry downs is going to be stored there as well. And I can actually send you a more commented version of this too. I'm realizing I didn't comment some of these variables here, so I apologize about that. So I'm going to run that, and that is going to find this JJ1 here is the count of how many dry downs it found. It found 56 dry down events within that 6-year time span. There's probably more rain events than 56, but this is finding kind of the long, the week-long dry downs that SMAP can detect. I'm sure there's some individual rain events in there that are, one day and then it rains the next day. It's not going to pick up on those and the short, maybe couple-hour dry period, just because the satellite can't sense that. So it found 56 drying events that SMAP can detect. So I'm going to plot now all of those dry down. I'm going to go back and plot. So I'm going to jump back up here. I plotted before. I showed this plot here already. This is just the scatter plot between soil moisture and the vegetation water content data. I'm going to plot now only the dry downs. Only those events when it rained. And I'm going to show little trajectories there. So there you go. So this is showing the red dot is the beginning of the dry down event, and the black is the remainder of the dry down event. So you can kind of see a little bit of a pattern emerging there. And one of the interesting things about it, and what did we do? We made soil moisture always dry. So this plot, what it's showing here is soil moisture always starts at a high value and it's going to a low value. So it's always starting at the right and it's moving to the left in time. So all of these are showing, so start here and this guy all the way over here on the right. Soil moisture jumped up to a very high value. And then as it dried in time, vegetation water content was going up. So in this dry location, we're seeing this behavior where soil moisture is going up after a rain event. And then you get over here to the left side of the plot, and it's a little hard to see what's going on. But soil moisture is now starting to decrease. So if it rains only a little bit, vegetation water content and the soil moisture both dry down together. They're moving below 0.1, a value of 0.1. They're mostly both decreasing. So they're both drying together. But if you get above the value of 0.1 there, they're kind of, as soil moisture is drying, you're getting an increase of water content in the plant. So this is showing, here's this kind of evidence then for this pulse reserve behavior. There's water going into the plant over multiple days, and it's mostly happening when it's wet. In this pixel, it's happening when there's this negative relationship here of water content going up while soil moisture is drying. So that's very simply what this script is doing. I think a lot of the minutiae here is just kind of finding the peaks and valleys of the soil moisture dry down, identifying the dry down event. And then you can kind of do some other fun things if you end up looking through that paper, and you don't have to do that, but one of the things you'll see in there is we calculate a threshold, a soil moisture threshold. I'm just watching the time here because I can actually go a little bit into detail on some of these pieces. But what this does is it actually calculates a soil moisture threshold there. It calculates that threshold at which, I'll jump back to this spot, the threshold at which you see this overturning happen. Somewhere around a value of 0.1 there, soil moisture is, and vegetation optical depth and the water content of the plant are decreasing. But when you're above 0.1 or so, that water content in the plant is increasing as soil moisture is going down. So when does that overturning event happen? There's a little bit of a non-monotonic little hump there. What is the kind of point at which you go from increasing vegetation water content to decreasing vegetation water content? And I'm just going to play this real quick so you can see what I'm talking about. So this found that hump there, that script. I can go through it in a second, but what it did, I'll just show you what it did and I can walk through it then. But it found approximately where that hump peaks. So somewhere above about 0.13 or 0.14 value of soil moisture. That's when the overturning happens. That's when you go below 0.13. That's when the vegetation starts drying, really, during that drying period. So that's kind of when water stress starts happening, basically. That's the threshold. So I can walk through that a little bit there. So what this is trying to do is draw that line that you saw through the data. It's trying to draw that line through the data. It's trying to find the median vegetation optical depth at each soil moisture value. And it's finding where the peak of that median vegetation water content is. So if you walk through this, it loops through each dry down, and it basically sets up values of vegetation water content at each value of soil moisture. So there's a little bit of and you can do this in different ways. This is the way we chose to do it. So this part of the script right here is really just our way of trying to find that peak of, when that overturning happens between drying and wetting. There's a little bit of uncertainty in there, but we drew it in a way that we found, we interpolated all of the vegetation water content data to specific soil moisture values, and then kind of conditioned on each soil moisture value and averaged across them. So just showing you what I'm talking about. It's a little hard to explain without looking at the picture. But we basically said at each value of soil moisture, a value of 0.1, what's the median value of vegetation water content? At a value of 0.2 soil moisture, what's the median value of vegetation water content? And just did that for each iteration. That's basically what the script is finding. And so it can draw that black curve, that black hump there. And it finds the median at that dashed line. So how do we find the median? So this finds the hump. This all up here finds that solid line there, that hump that we saw. And the rest of this down here then is going to find the peak. So it takes the median over the vegetation water content data set for each value of soil moisture. And then where is it here? There it is, this line right here. Line 168, it finds the peak of that smoothed curve there, and that's the threshold. That's the threshold of soil moisture. So that's where, that's the soil moisture value at which water content is decreasing. So this is the way we've done the analysis, and I'm sure there are other ways to do this. It might be might be a better way. So I'm gonna stop there. There's another part of the script down here that looks at the strength of the pulse response. And I'm not really I don't need to walk through that. It basically just shows the strength of how quickly and how strong the increase of water content is. And it actually does a bootstrapping procedure to calculate uncertainty of how strong that increase is. So it does a statistical method to calculate the uncertainty of how strong the water content response is. And I can just explain real quick if I go back up to this plot right here. Nope, sorry. This plot. That metric is going to calculate how big the slope is of the increasing water content, vegetation water content, per decline in soil moisture. So that's assessing how strong the vegetation water content increase is in this pixel. And I'll show you after all of this, I know I've kind of walked through the weeds of one pixel, but I'll show you what each pixel looks like in the pictures. Then you'll have a little bit of an idea of the spatial distribution and where this phenomena happens and where it does not. So I take any questions now? I think that's pretty much it for the coding part. And yeah.
Yeah, so a couple of quick questions. One, that's a very interesting graph actually. And is it possible to tease out plant water storage from that particular curve? Because it just seems that initially when the soil moisture is declining, but the plant water availability, not the plant water availability, but I think the plant water itself is increasing. I think, is it possible to somehow gauge at what could be the plant storage? Because after that, it just seems both are sort of declining.
Yeah, so the y-axis there is effectively the storage. So the y-axis there is the level of storage of water in the plant. The delta, how much water content is changing from right to left on this diagram, is effectively how much of the storage is changing, how much water is going into the plant. Does that answer the question? So yeah. The y-axis is effectively the water storage in the plant. What this is showing is the time evolution of that water storage over a couple days after it rains. So each of those lines is like a 10-day period after a rainfall event. So if it's kind of moving up from right to left, if it's going upwards, kind of, if you look at the top one there, it's going up. That's water going into the plant effectively.
And these are from, I assume, SMAP, right? Both of them are SMAP.
Yep, 30-kilometer. This is a 30-kilometer integrated data set. So the size of Eastern Massachusetts, or if you're all in College Station, you're probably going to get a size from College Station down to like Houston.
Yeah. Another question, very interesting, is are you able to distinguish between vascular plants and non-vascular plants, like Moses or bryophytes? It's a very deep question.
Yeah, that's a good question. There might be a way. The issue is because we're at such a coarse resolution, you'd have to go to a higher resolution data set to actually look at that, I think. So you want to look at actually radar is another method. You can get this data set at a higher spatial resolution and 15, 20 meter, 30 meter data set. And then you might be able to look at a place with more mosses, like a non-vascular plant, and see if those dynamics are different. At a 30-kilometer scale, I think you're going to mix things too much. But that's a good question. And yeah, I would love to be able to do that. That'd be pretty cool.
So I guess, what's your opinion if we, right now we are looking at the SMAP data and it's giving us an effective value. What's your opinion, because you have worked on emeriflux data as well. Do you think, one, you can see the similar sort of graph where there's a peak and then there's a decline? And two, will that be able to help us in sort of distinguishing between different plant species response?
So yes, actually, some of the data sets I showed you in the beginning of the slides there were from carbon fluxes. So that was some of the station data there. So you would be able to see that in certain plots. I think that question, the second question of the kind of species dependence, I can answer with the slides coming up here. So I can answer that after we jump off of Python here. But the answer is yes, we can start to think about different types of vegetation and how they look on that curve. Okay. Yeah, between shrubs versus grasses, not really, shrubs versus grass versus trees, that kind of resolution, but not different types of shrubs. Just we're integrating too much area. It's too much. We're kind of averaging tons of different things within a pixel. So I think from here, I'll jump off of Python. I'll get back to the slides. So the Python script available, it's maybe simpler in nature than what a lot of other folks are going to be doing, but I've used this script, and it was partly written by my PhD advisor, and I've adapted it and kind of built on this a lot over the years. It's funny how this script kind of keeps coming back many years later. I'm still using it. I adapted it from MATLAB into this, I think a couple years ago. So yeah, I think a little bit simpler than some of the coding some of the others will show later in the week. So I'm going to jump back to these slides and answer some of those questions. And so that's the analysis. It's on Python there. We showed you one pixel. What happens when you run all the pixels? Okay. Can you see my screen? No, you can't. Now, can you see my screen? Back to the slides?
Yes.
Cool. All right. Okay, so this is a pixel in South Sudan. So this is very similar to what I showed you in the Python script here, but it's now showing you a little bit more clearly with color. And so if you clean up that Python script and you make a nice plot, you get this plot here. So the soil moisture time series is in blue, and that vegetation water content time series is in green. And again, we're identifying all those different dry down events, the dry down events in the red, and now we're looking at the water content from the plant. And starting to try to identify emerging behavior from it. And so, yeah, we're picking out some dry downs just like we did, and then plotting it, just as I was explaining there in that scatter plot and some of the trajectories there. But this is the general idea that there's a dry down progression in time from right to left on that diagram. And you can kind of see soil moisture going up and then down. And so right here, this is all the grasslands. There's, 100,000 interstorms. They're integrated there on the left. And in the paper, we found a way to average out and plot each one in a kind of easy way to look at. And so that's the behavior on the right there. Those are those individual trajectories that we averaged together over an area in South Sudan to show that increasing water content behavior in wet conditions and then decreasing in dry conditions. And to answer your question, Devasish, this plot looks a lot different in different types of vegetation. For trees, you might see actually this increasing behavior might not happen actually. Sometimes they don't always respond. They don't have like a growth behavior every time after each rain event. So that's one example there that you might not see this type of behavior in a forested area. You might just see things starting up on the top right of the plot and all moving down to the bottom left, kind of just linear, just goes down, it decreases. They lose water together. So this plot right here, then coming to the finale of all of that, if you run that algorithm on each pixel, and I'm doing this here for Africa, South America, and Australia, the red is all of the places, and the red is the strength of how quickly that vegetation water content is going up after it rains. So dark red, I know it's a little unintuitive here, but the more negative values, dark red means that water is going into the plant very quickly and really fast. It's even growth is happening. It's a really big, so you can see in Sahel in Africa, southern part of Africa there, Mozambique, Namibia, Botswana, really high sensitivity of plants to rainfall events, especially in the wet season. And what do all these places in red have in common? They're all dry places. So the kind of big point of this whole paper was that dry places definitely, in accordance with that theory, show an increase of water content after it rains based on this data set. And that there are other places, like wetter places don't show that behavior. If we go into the Congo Basin here, and Congo Basin on the right, the Amazon Basin on the left there, there's no data there because it didn't find this behavior at all. There weren't really dry downs to look at, and even when there was a dry down, this behavior of increasing water content after rains did not happen. So really wet places don't show the behavior. What's interesting, I think, about this paper that we saw that we didn't expect was that so much of the landscape does show this behavior, much more than what Emmanuel Neumayer hypothesized. So it's actually a little bit dry places. Places are just a little bit dry, even into like the Great Plains, a lot of the grasslands in the Great Plains, croplands throughout the Midwest do show this behavior to some degree. And then they're not very dry. It's not a desert. It's not that opportunistic of vegetation, but here it's showing that even if you go into kind of dry tropical forests, you still see this behavior a little bit, in a little bit humid behaviors, in a little bit humid regions, you still see this behavior. So it's a widespread behavior. What I'm saying here is across the globe, about half of the landscape shows some response to individual rainfall events, and some growth and some rehydration. Individual rainfall events do matter to each plant in a lot of places across the globe, to a lot of plants, I should say, across the globe. So that's a kind of one of the big conclusions. It sounds very trivial again, but it's something we really haven't thought about, and the theory didn't really match up with the data that we found like this so far. It's not only just desert, really dry species. And here's the Western U.S. that same kind of plot here. Sorry, the full U.S. the same plot. The blue in this case, I'm sorry I switched the axis, but the blue in this case is everywhere where we see this behavior. So in the Western U.S., Colorado, you get a little bit into the Great Plains, you see this. And this is actually a different metric. This actually does extend further east. So I don't know, I grabbed the wrong plot here. Sorry about that. The bottom left plot, to answer your question, Debisish, that is using those little dots there. That is showing crop, sorry, getting tired here. That's showing the carbon flux data. So it shows also the carbon fluxes show this response as well, that drier locations have a stronger response to individual rainfall events, but even wet places still do show a response as well. It's not nothing. So, you go to the Great Plains and kind of transitional regions in the middle of the US, you do see you do see a big response to individual rainfall events. So it becomes progressively weaker as you go into humid regions. It doesn't just drop off. It's a continuum. And it's supported, I think that the bottom left plots show it's supported by in situ data to some degree from the carbon flux data. So it's not, the satellite's not the only thing that shows this. So there's some believability in that. And if you want to kind of argue about how well the satellite does at picking up this response and error in the satellite data, that's a whole other conversation. And it's something I've thought about a lot. But we're pretty confident that this is not noise from the satellite. I talked a little bit about growth versus non-growth. Is growth happening in a separate study? I'm not going to go into great detail on this, but we did find instances that growth was indeed part of this response. So I mentioned early on that vegetation water content, if it goes up, it's not clear if it's plant water if it's not clear if it's just the water in the plant, the kind of saturation level, or if it's a growth that more vegetation's been added. Well, actually, if growth tends to happen after very large rainfall events. So when you have a very big rainfall event, you get a lot more growth. And that kind of comes back to this original plot here I showed from a Colorado grassland, that where they dumped a ton of rainfall on the surface, there was a lot of growth. This does happen actually from the satellite data, but you have to have a big rainfall event to see that. And we were able to corroborate this with a leaf area index product. Actually, it was a geostationary satellite data set. It's a little bit of a tangent to get into that of how we detected the growth, but there is a way with a different geostationary satellite data set that's just located over Africa for this case, that shows minute measurements. So there's a way to back out if there's growth happening from that data set. Okay. So how many slides? Okay, this is the last couple slides here. So I'm going back then to the beginning. What does this all mean? Why do we care about this? I looked at individual rainfall events. Who the heck cares? Why does it matter? So the top point, that pulse reserve hypothesis that this person, this ecologist talked about in drylands, it doesn't just occur in drylands. It's strongest in drylands, but it holds outside of drylands. As you move into wetter ecosystems, we do see growth responses. We do see strong hydraulic responses in plants. So that hypothesis is true, but it was limited in how much of a scale that it gave to it. Why does some of this matter then as well? So for modeling, why does some of this matter then for the global carbon cycle and what we're doing to model ecosystem function? And what have we learned from these pulse response behaviors? So terrestrial biosphere models are ways that we can predict. You can use them to predict crop yields. If you kind of bring in crop information to them, you can use these terrestrial biosphere models to predict how much carbon uptake is happening. You can make predictions about the carbon cycle with them. They're pretty powerful models. They're really a lot of folks work on them. They're pretty well accepted in the community. But one of the things they do is they use these stress functions. And what the stress functions do is they model how much GPP, this I should have put on the y-axis here what GPP is. This is photosynthesis. This is how much photosynthesis a plant is doing. These models prescribe how photosynthesis should respond to soil moisture based on what we know about them. And they use these very simple curves here, that red and the black line here in the middle on the right. Are you able to see my cursor at all when I'm moving this around?
Yes.
You are? So that's a type of curve they use. They use beta functions or these S-shaped curves to describe how soil moisture influences photosynthesis. But what we've learned actually from this satellite behavior is drylands and even into some, slightly wetter ecosystems look a little more like this. There's an hysteresis. They kind of make this loop-de-loop. And so right here, these terrestrial biosphere models, the stress functions that they use to describe how plants respond to soil moisture are partly wrong, especially in dry locations. So there's going to be an error there in their photosynthesis function for the annual amount of rainfall that they pick up in drylands, and then even some wetter locations, subhumid locations, I'll call them, that are more transitional. They will probably have an error associated with this. So that's one of the things we've learned from this. This is a complicated figure. I'm not going to go through all of it, but what this shows, drawing the box around this, plant response sensitivity to wet days. This was a bigger analysis that we did for a global analysis for how plants respond to fewer larger rainfall events. What this says is that plant response sensitivity to wet days, that figure I'd showed previously, how much, how sensitive plants are to individual rainfall events, matters for scaling up for annual timescales. So this just says that if I know how much a plant responds to an individual rain event, I can tell you something about how much it's responding over an entire year. That's what this is saying. I'm just showing a plot to show that I did that. It's a little hard to show with all the statistics in the background. I haven't found an easy way to show that for you. But what this says, and I'm going to go all the way back. I should have copied it back onto here at the end. But what this says is that there is a reason why, when I asked this question before, how would larger storms but longer dry spells influence plant function? We all said we kind of said mixture to mostly decrease. Why is it that dry locations are mostly positive? So coming back to this question here, why are dry locations showing mostly positive responses? It turns out that they really like larger rainfall events, as we've shown from this whole thing and the Python script, that dry places, plants there are really opportunistic. They really, really, really like that big rainfall event. And so if you have fewer but larger rainfall events, they're going to say, okay, I'm willing to get rid of a couple rainfall events throughout the year. But if you give me a big rainfall event, I'm going to really use that. So that's why you get mostly, more positive, plant response sign when there's fewer larger rainfall events. So that's what that figure said in the end there. I'm going to jump to the last slide then. So yeah, what kind of revisiting these questions I had in the beginning, how prevalent are plant water content responses to pulses of water availability? Mostly in drylands, of course, but they're very prevalent. In that specific study, we showed about 50% of the tropical domain. If you extend it across the globe, it's about the same number. So about half of plants across the globe do respond to individual rain events. In the other places, I think they care a lot more about seasonality. They care about seasonality of light, seasonality of temperature, and such. And yeah, I think the hypothesis here is partly wrong. It's correct for drier locations, but there's still a response, but decreasing strength of response in wetter locations, and it eventually kind of disappears once you get to very humid places like the Amazon and the Congo rainforest. You're not really going to see this at all because it's not water limited. They care about light and other factors. So individual rainfall events don't really matter there. Maybe you probably figured that. So I'll probably end there, but I think I kind of want to end on a note that we walked through this idea of individual rainfall events, and I kind of told you at the beginning it's kind of sounds like a boring topic, but you can actually start to use that to predict a lot of things just from that little piece of information there. And then when you grab satellite data, we're now in a data-rich time period. We have satellites in space. When my PhD advisor was doing his PhD in the same lab that I was doing it in the 80s, they only had models, and so they had to kind of guess at a lot of the behavior based on individual field experiments. A few field experiments out there. They were doing a lot of guessing, and then they were modeling based on theory. We have data now, so we can go out and actually test these hypotheses, and do this with some interesting things like microwave data. So we can actually look at hypothesis from the 1970s and say, is that true or not? And does it matter? And it turns out that some of these things do matter. And, I thought this didn't matter at all. And I kind of changed my mind over the years. So I encourage you all to do something cool with data as well. And, we're in a data-rich period. And even now, 6 years after my PhD, or a couple years after my PhD, and if you're all doing that now, you have even more data than me. It's an exponential growth curve. So go do something cool with data.
Okay, thank you, Andrew, so much. We have a lot of questions, but before that, before I forget, I'm just sharing the link to the feedback form. Please go to the feedback form and put in your response. And then let's come back to the questions. There are so many interesting questions, and I think what we are going to do is go through it in a sequential fashion. So the first question is, are you able to visualize ecotones or areas of transition between different biomes? And I think there's a follow-up, and do these types of corridors affect data metrics, and are they accounted for? Yeah.
Yeah, good question. So in the context of this problem, I'll jump back here. In the context of this problem, we used very rudimentary vegetation type data sets to pick out areas like the Sahelo-Sudan grasslands. And then we looked at shrublands and then croplands in different parts of Africa, and partition them. And they do show different behavior. They're pretty basic. And so kind of a broader answer to that question is you can get better ecotone data. You can get better vegetation type data out there to start to look at differences of these. More generally answering that question, not in the context of this problem, there are better data sets out there than what we did to partition out the ecotones and the biomes. And as those data sets become more available, like hyperspectral data, we can start to really identify vegetation types. I think you'll see a boom in better identifications of ecotones, but we used very crude ones here, and the answer is yes. We did see some differences in the crude ecotones metrics that we used. Hope that answered that.
Yes. So the next question is, so is this, I'm not particularly sure what behavior, but is this behavior common to all diverse flora present in the dry areas the dataset refers to? Could the biology difference between desert plants and tropical plants to be a confounding variable affecting the interpretation of water absorption rate?
Yeah. So that's the limitation of this data set being so coarse. And I'll jump back up here, kind of just put this as the background as I answer the question. This data set's pretty coarse. And so we're averaging over many types of vegetation within each pixel. And so the answer to that is in these pixels we looked at, most of the vegetation there, as we integrate over it, did this behavior. But I can guarantee you, if you go zoom in and you redo the analysis with an imaginary 10-meter data set that has the same properties, I'm sure you're going to find a lot of variability within that pixel in dry places that shows that there are some plants that do not do this behavior. But what this shows is that most plants do this behavior in that pixel, or at least the ones that pop out in the signal do this. Yeah, a good question, and we're limited by data there. Yeah.
Yeah. So moving on to the next question, considering the coarse resolution of SMAP, for a lot of landscapes, this could include multiple landforms like uplands, riparians, or different soil orders. What is the data effectively telling us? What landscape contexts are the best or optimal use of this data? And maybe I can add on to that a similar question is how well does SMAP perform in very humid systems versus arid systems?
Yeah. Oh, man, good questions. You're making me think here. So the first question about where does this work best? The reason I'm showing you Africa and the reason we did a lot of African studies is it's such a big continent. It's huge. It's a huge continent, and it's pretty smoothly varying. The way Africa, it's centered at the center, pretty much on the equator, and at a spot where as you move north and south, the climate kind of it has a simpler gradient of change, and there's a lot of kind of homogeneous landscapes. So it's actually a very good place to use SMAP because there's a lot of kind of homogeneous landscapes. There are mountains and there's a lot of riparian zones, but on average, Africa is easiest to work with this data set. For the Western US, it's not good. I mean, the Western US is mountains, and if I mean, if you're in college, a lot of you are in College Station. If you drive west and you go across New Mexico and you go into Arizona and you go into Utah, the landscape changes very quickly, very, very quickly. There's a lot of terrain changes. You're going up and down. The algorithm does not do well with angles of topography. If there's very big angles, it doesn't account for sloped surfaces very well. We're not sure how much that hurts our analysis. Actually, that hasn't been really well evaluated for soil moisture. But yeah, I mean, the Western US is not going to be a great place to use this data set. So you want something higher resolution there. Africa's a little easier. Will this data set work better in the tropics versus dry places? I think is that the question? Will it work better?
Yes.
More of a question about if there are trees or not, rather than the kind of humidity level. Trees. So the microwave wavelength we're talking about here is 20 centimeters at this specific frequency that we're dealing with, L band, for vegetation water content. 20 centimeters, and what that means is that wavelength is going to go through whatever's smaller than 20 centimeters. So if you have a cloud, you have a lot of small water particles, you're going to go through that. If you have a wall, you might go through that. Vegetation like a trunk, if it's a really big trunk and you have a lot of branches and trees, it's not going to go through that. So this data set does not work well, and the soil moisture data sets out there do not work well in forests. It's a pretty known issue. You get a lot of cloud noise when you look at the data, and it's very noisy data. because you're getting a mixture of soil signal, a little bit of a soil signal, you're getting vegetation scattered signal. So if there are a lot of trees in a pixel and you're retrieving soil moisture, water content vegetation, it's not going to be as good. It's just bar none. It's not going to be as high quality.
Interesting. Yeah. Uh, Another question is in the graph comparing soil moisture and VOD on the y-axis, could you explain how the 4 points on each of the graphs were selected? Maybe it's the graph that,
Yeah. This was the graph that I showed in the Python script, maybe.
yeah.
Then it could have even that's here. Well, I'll just show one. How were they selected? I hope I'm thinking of the right. Hopefully, I'm aligned with the question. The person who asked the question here. Hopefully, I'm on the same wavelength as you. But the Python script goes through and it finds the peaks and the troughs. It finds the highs and the valleys, and it finds the peaks and the valleys that have at least a couple data points, like 3 or 4 data points between them. And it has to have at least 3 or 4 points between them, and the soil moisture data must always be decreasing. If there's a choppiness, if it goes up and it goes down, see if you go look over here, it's up and down, up and down. There's no dry down there that we can really detect. Whereas if you go over here, you see a nice decline over a couple days there. Those 2 criteria are met, that there's consecutively decreasing soil moisture and that there's also a couple days of decreasing soil moisture as well. So that's how we kind of pick out a rainfall free period that we can do our analysis on. I hope that makes sense, but that's what the Python script is really doing at the heart of it.
Okay. Another question is, what is the earliest data we have of VOD? And do we see any difference from today's graphs that could be attributed to climate change or some other factors?
Yeah, so I'll go back to this slide here. So the 2 top ones here are L-band. These are available between 2009 for SMOS and then for SMAP 2015. So with those specific data sets, really climate change signals are difficult to detect. SMOS maybe a little more now. We've got 15 years. AM series, this is a little bit higher frequency of a wavelength for vegetation optical depth. We can start to piece these together, but we don't get as far into the canopy. So what I'm getting at here is you can combine data sets, and there's folks that have done a very good job combining data sets and actually used old satellites that weren't, I would say they weren't as set up for such a problem, and they were a little bit noisier. But those ones go back into the 1980s. They don't have the same setup as what SMAP and SMOS do, but you can get a vegetation water content data set from those satellites. And what these folks have done is pieced together SMAP, SMOS, AMSR, and then went all the way back to 1980. And they made one conglomerate data set of this vegetation water content. How this specific behavior changes in that time series would be tough. I don't, not tough. People haven't done it. I haven't done it. I haven't looked at it and how that might change over time. But people have used that to look at just trends in the water storage and vegetation and saw different interesting things, like how much loss of vegetation there is between 1990 and 2020 in a certain region. And some papers are showing, hey, since 2015 we've seen a loss of biomass in the boreal forests. There's a loss of this vegetation water content there that's maybe due to disturbance. So those are types of questions you can use for that. But yes, there are ways to look at that type of a question, albeit with a little bit of work in between.
Okay. And maybe it's good to end on a last question, which I had, because you started talking about how using NDVI products, we just look at the surface, because it's just infrared and it cannot penetrate. But if you look at the vegetation water content algorithm from SMAP, it uses NDVI as the input, if you look at that. So according to you, because you have been working on this and you're the right person to answer, I don't know whether the VOD algorithm is different, but at least the VWC algorithm that was done by Rajat Bindlish and others, it just it uses NDVI. So are we not using the data as far as variability is concerned? The NDVI variability percolates into the variability of vegetation water content, or are we able to achieve a more something more than that?
Yeah, so that's what this data set's doing. So the reason, I mean, I didn't get into explaining it, I thought it was a little bit too detailed, but this multi-temporal dual channel algorithm, what Alex, the previous PhD student before me, and then my PhD advisor had previously done was found a way to retrieve water content data without NDVI and did it in a way that reduced noise. So yes, that data set that they have for that SMAP official data product, they use NDVI, and that is a flaw for this type of analysis. You probably would be doing this analysis in error if you're using that data set for this. That's kind of why we went on our own and made this own algorithm to back out vegetation water content without without the other assumptions and using other data sets like NDVI that would guide it maybe in the wrong direction for this type of analysis. So that's a great question. And yeah, folks spent years doing just that. It's not a trivial thing to write this script for this MTDCA algorithm. But yes, we've done that and thought a lot about that question. It's a really good question.
Okay, so thank you so much, Andrew. I think it was really interesting, and I'm pretty sure everyone must have learned a lot. So let's thank Andrew for the last time, and then if your question was not answered, please feel free to reach back to us either through the feedback form or through directly emailing me. And last but not the least, I actually forgot to acknowledge Rishabh, who is helping me here with all the things. And thank you, and see you all guys tomorrow.